All right, you guys, so with your notes packet, I want you to just look right here at this area where it says DNA structure, okay? So I'm going to just go over review a couple key terms that you can add in this space, or once again, you could do it on your own sheet of paper, whatever is easiest for you. Okay? All right. So when it comes to our DNA structure, okay, you could have something written down. Hopefully you have something written down as far as who pairs up with who. Okay, your base pairing rules, A's with T and C's with G. You could also put down the term anti-parallel. Okay, and this is important to understand the direction that DNA moves when it gets replicated. And so I kind of broke down the term for you. Right? So once again, this can go in that section of your notes, or you can put this on your own. But anti-parallel basically just means right here, when two molecules are side by side but run in opposite directions. So they're side by side, but they're opposites. Okay? The two strands of DNA are anti-parallel. The head of one strand is always laid against the tail of another strand. So I like the picture right here because this is a perfect example of why they're called anti-parallel. Okay? So here's a 5' end, and there's a 3' end. So here's the head, and here's the tail. Okay? And the head is going to be laid down and match up to the tail of the other end. Okay? Just like the tail is matched up to the head of the other side. So they are anti-parallel. Okay? And you can kind of see here in that DNA double helix, when it comes to the 5, matches up over here to the 3, and you follow that on down, and 5 matches to the 3. Okay? So that's a new term for you. We'll kind of reference that a little bit more. I liked this picture in this DNA double helix. Once again, it's showing you. All right? So it's reference to where like that final oxygen molecule ends. But you can see here's a 3' end, here's a 5' end, here's a 3' end, here's a 5' end. So they're always the opposite. Okay? Once again, anti-parallel, the way that they run. All right, so what I'd like to do right now is we're actually going to jump to a video. I don't know if you guys have ever seen the Amoeba Sisters. They do a fantastic job. And so we are going to watch these guys. Let me make this a little bit bigger so you can see it. And then we'll go back into filling in our notes here. All right, let me get this started. 1 second. All right, here's a video. You could write down things if you'd like, but for the most part, I really just want you to pay attention and listen to their explanation of how DNA replicates and why it's important.
DNA. We talk about it so much. It is the ultimate director for cells, and it codes for your traits. It's a major component of what makes you, you. When you have a really important molecule like DNA that is ultimately responsible for controlling the cell, it would make sense that when you make another cell, like in mitosis, you would also have to get more DNA into that cell. And that introduces our topic of DNA replication, which means making more DNA. First, let's talk about where and when. First, where. It occurs in the nucleus. If the cell has a nucleus, remember, not all cells have a nucleus. This video clip is actually going to focus on the types of cells that do have a nucleus. They're known as eukaryote cells. Prokaryotes, which are cells that do not have a nucleus, they do not have a nucleus. They do things a little differently. They also do DNA replication, but that is not going to be our focus for this clip.
All right, so just kind of backing up a little bit. Eukaryotic cells, okay, those are animal and plant cells. Where prokaryotic cells, that's your bacteria. And so, like we said, we're going to talk about DNA replication that occurs within animal and plant cells.
Next, when. When does this happen? Well, this typically happens during the stage known as interphase. Interphase is when a cell is growing, it's carrying out cell processes, and it's replicating its DNA. You know what it's not doing at the exact same time? Dividing.
All right, so another thing that I want to pause here really quick is I just want to talk about what they reference there about interphase and S phase. So I just have a little notebook I'm going to pull up here and just do a little bit of writing. Okay. So when it comes to the cell cycle, and they reference interphase, okay, so your cell goes through these different stages as it begins replication. So this is all about taking a DNA double helix. That's awful. Taking a DNA double helix and creating, let's see if we can do better. Oh, so much better. And creating 2 DNA double helixes, right? So it's taking one and then making 2 from it. So we're replicating our DNA. So what you have here is a phase called G1. All right, in G1 phase, this is when the cell just grows. And this is things like organelles within the cell are replicating, and it's just maturing and getting bigger. Okay, and then it moves on to S phase, and this is actually the stage when your DNA copies. Okay, so this is where in your cell you have 46 chromosomes. Well, now that's doubled, right? So now you've got 92 chromosomes. So everything's replicated twice. And then it moves on to what we call G2 phase. And this is where more organelles replicate. So once again, the cell also just continues to grow. Okay. And then we move into M phase. And when it comes to mitosis, this is where you actually get division of the nucleus. So if this is your cell, it had 1 nucleus, now you have 2 nucleuses. And those nucleuses have each an identical copy of DNA. Right? Because when did we copy that DNA? Well, we copied it in S phase. And then finally, it ends at C phase. And that's where this cell will actually split into 2 new cells. So you have 2 new cells, each with their own nucleus, identical to each other, because once again, we copied that DNA in S phase. And then what's going to happen? Well, it's a cycle for a reason. It just starts right back over. Right? So 1 cell will end up making 2 cells with identical DNA because we're replicating that DNA. So this is the phase. Ooh, let's highlight. This is the phase that we're talking about right now in DNA replication, is this S phase. Okay. All right, we're going to go back to the video. Let me pull that up. Okay, so we'll return back to our video.
You don't want a cell to be replicating DNA and dividing at the same time. It's a little bit too much multitasking. So DNA replication does not happen during cell division.
All right, now here's a new term called interphase. And interphase really is the combination of those first 3 phases that I told you. G1, where it grows, S, where the DNA copies, and G2, where it continues to grow. So interphase combines G1, S, and G2 all together. And then there's that mitosis, where the nucleus is actually splitting and making 2.
Otherwise known as mitosis. In fact, cells replicate their DNA before division processes like mitosis and meiosis. Because once you make a new cell, you better have DNA to put in there. I think DNA replication would actually make a great video game. It's actually quite exciting. I'm going to introduce the key players in DNA replication so that you can get some background information. Now, the majority of these key players that I'm going to introduce are enzymes. In biology, when you see something end in ASE, you might want to check as it's very possible that it may be an enzyme. Enzymes have the ability to speed up reactions and build up or break down the items that they act on. So here we go with our key players. First, helicase. This is the unzipping enzyme. If you recall that DNA has 2 strands, you can think of helicase as unzipping the 2 strands of DNA. Helicase doesn't have a hard time doing that. DNA polymerase, the builder. This enzyme replicates DNA molecules to actually build a new strand of DNA. Primase, the initializer. With as great as DNA polymerase is, poor DNA polymerase can't figure out where to get started without something called a primer. Primase makes the primer so that DNA polymerase can figure out where to go to start to work. You know what's kind of interesting about the primer that it makes? actually a piece of RNA. Ligase, the gluer, it helps glue DNA fragments together. More about why you would need that a little later. Now don't feel overwhelmed. We'll go over this sequence in order. Please keep in mind that like all of our videos, we tend to give the big picture, but there is always more detail to every biological process we cover. That's really true for DNA replication.
Okay, so just a couple of things. I love that it brought up these enzymes, and they each have a very unique role in creating our DNA double helix into two. So the first enzyme was the one that would break the DNA apart and break apart those hydrogen bonds, and that is the helicase, right? H, helicase for break hydrogen bonds. HH, helicase, hydrogen bonds, breaks it apart. And then there were some extra enzymes that they talked about, and they're actually not ones that we're not going to learn every single enzyme that plays a role, but it did talk about the primase that brought in a little start spot for the larger enzyme to come in, and this is what I want you to know, and that's that DNA polymerase. So that enzyme comes in, and it adds nucleotides according to the base pairing rules, A with T and C with G. Okay? So helicase breaks apart the hydrogen bonds, and that DNA polymerase adds nucleotides, okay, according to the base pairing rules. And also remember that polymerase has to go a certain direction. It goes from the 5' to the 3'. Okay? And then the last enzyme that we're going to have you learn is the ligase. And the ligase is what glues the new DNA strand to the old strand. Okay? Those three major enzymes.
DNA replication starts at a certain part called the origin. Usually this part is identified by certain DNA sequences. There can be multiple origins within the DNA. At the origin, helicase, the unzipping enzyme, comes in and unzips the DNA. SSB proteins, that stands for single-stranded binding proteins, bind to the DNA strands and they keep them separated. Primase comes in and makes RNA primers on both strands. Now remember, that's really important because otherwise when DNA polymerase comes in, it wouldn't know where to start.
Right.
Now DNA polymerase can get to work. Remember, it's the important enzyme that adds DNA bases. Now, you have two strands, right? They're not identical. Remember, they complement each other. They also are anti-parallel. So that means they don't really go in the same direction. With DNA, we don't say a strand goes north or south. The directions for the DNA strands are a little different. We say that DNA either goes 5' to 3' or 3' to 5'. This can seem very confusing. What in the world does that mean? Well, the sugar of DNA is part of the backbone of DNA. It has carbons. The carbons on the sugar are numbered right after the oxygen in a clockwise direction. 1', 2', 3', 4', and 5'. The 5' carbon is actually outside of the ring structure. Now you do the same thing for the other side, but keep in mind the strand is flipped, just because DNA strands are anti-parallel to each other. So let's count these. Again, clockwise from the oxygen, 1', 2', 3', 4', 5', and the 5' is out of the ring. This strand on the left runs 5' to 3', and the strand on the right here runs 3' to 5'. Well, it turns out that DNA polymerase can only work in the 5' to 3' direction. So the strand that runs 5' to 3' is fine. It's called the leading strand. But the other strand will make it a little tricky. DNA polymerase can only go in the 5' to 3' direction. So RNA primate...
All right, so we're actually just going to pause it there because the biggest thing that you need to understand is those three enzymes that help create the new DNA double helix, and that it's always, that polymerase is always going to run the direction of 5 to 3, and these strands are going to be different from each other. So this polymerase is going to go this direction, okay, towards the replication fork, but this strand is going to go this direction, away from the replication fork. So it can just, as it gets separated, it just adds new strands, right? So don't necessarily worry about the terms